All right, so in our last chapter, we talked about asexual reproduction, where cells give rise to other cells, and then what goes wrong when cells don't replicate properly. Today, we're going to talk about meiosis and sexual reproduction. So we're going to talk about the basics of how we see unique individuals being made through the act of sexual reproduction and why that can be good, and compare this with mitosis a little bit more. So if you're not feeling super confident with mitosis, do make sure that you go back and watch that lecture video and then come back to this one, okay? So as always, here are our chapter objectives. I know it looks like a lot, but really what it is it's talking about the differences between mitosis and meiosis and a lot of the vocabulary here. So remember, this particular unit is very, very vocab heavy, and that vocabulary carries over through the semester. So flashcards, build them on Quizlet. There's probably a billion of them built on Quizlet. Make sure that your, even index cards, whatever it is that works for you, make sure you're practicing the vocab, and then practice using it as much as possible as well while you're going through your homework or note-taking processes. If you're drawing out mitosis or meiosis, make sure that when you're writing out the phases, you're using the vocab as much as possible. So let's get into it, okay? So what is meiosis? Well, meiosis is sexual reproduction, but it's not really sexual reproduction, okay? Meiosis is the precursor, or what happens in order to allow sexual reproduction to reproduce a new individual that has the right number of chromosomes for that particular species, okay? So what do I mean about that? Well, we as humans have 46 chromosomes. We have 23 pairs of chromosomes, okay? We have 23 pairs because we are diploid organisms. Each copy of our chromosomes has 2 copies, so 23 times 2 gives us 46, okay? That is what makes us human. If you look at other organisms, they have different numbers of chromosomes. Mosquitoes have 6, fruit flies have 8. I think dogs have 72. I think there's a fern that has over 4,000. Like, the copies of chromosomes is dependent on the particular organism we're looking at, okay? Meiosis is a type of nuclear division then that actually reduces the chromosome number by half. So you are taking a diploid organism who has 2 copies of each chromosome, and you are reducing it to a haploid cell where it has 1 copy of each chromosome in that particular cell. Because if I took like human skin cells, they would have 46 chromosomes. If I tried to put 46 chromosomes with 46 chromosomes, I would not end up with 46 chromosomes, right? That is not how that math works. But if I want to make a new organism that has 46 chromosomes and I'm bringing together 2 individuals to do so, that is where we get that haploid number of 23, okay? The other thing that meiosis does that's actually really key to allowing individuals to be unique through the process of meiosis and reproduction from sexual reproduction is shuffling or mixing up of the genetic material of the individual. So, you share DNA with your siblings, with your parents, with whoever, but you're not 100% the same unless you're an identical twin, right? And identical twins are a little different because they're formed by the splitting of the cells early in development, and we'll talk about it a little bit. But when a cell is fertilized, so when an egg is fertilized by a sperm, okay, every single event is completely random. And so meiosis reduces the number of chromosomes, creates 4 unique cells that we call gametes that can then recombine through fertilization to generate a new individual who is genetically unique from anything else. So meiosis is really important in that aspect, okay? Meiosis isn't the act of making a baby directly. It's not the act of growing a zygote. When an egg is fertilized, it becomes a zygote. That zygote then switches to mitosis for that growth and development aspect. But meiosis results in a halving of the chromosome numbers and a shuffling up of that genetic material to give us a unique situation, okay? So let's quickly compare mitosis and meiosis, which if you did check your knowledge or check your understanding at the end of chapter 8, you should have already done a little bit of this by watching that Amoeba Sisters video. But when we talk about mitosis and meiosis, okay, one of the things that I really like to do in a seated class, and I like you to think about in an online class, is when I say, if I'm comparing something, I have to give both sides of whatever it is I'm comparing. So if I say that mitosis produces two genetically identical daughter cells called clones, I have to then look at meiosis and say, well, what's the equivalent of that for meiosis? So for meiosis, I'm producing four genetically unique cells called gametes. Still talking about the number of cells, still talking about how the cells are genetically, still talking about the number of cells, but we can then see the differences here. Okay? When you compare something, and it doesn't matter if it's for this class or any other class, make sure that you are thinking through the entirety of that answer. If you just said that mitosis is diploid, but then you didn't say meiosis is haploid, you're not telling me that you understand the full picture. Okay? A comparison is a full picture. So, if we talk about mitosis, right? Diploid cells, genetically identical to the parent cell, two cells at the end, we call them clones. It's used for repair, growth, and development. It's typically a fairly fast process compared to like meiosis and sexual reproduction. And one of the things that's really interesting is in a changing environment, individuals that solely rely on asexual reproduction as a way to generate new individuals in that population have a low survival in a changing environment. Okay, so I have my example here of bananas. Bananas that we eat today are not the same bananas that we used to eat. So the bananas we used to eat are why like Runts and banana flavored candies taste so different from the bananas you buy at the grocery store. Okay, that is more close to the taste of the bananas that we used to eat. So why don't we eat those bananas anymore? Well, that entire population of bananas was made from asexual clones. So bananas that we eat don't have seeds in them, okay? You can't plant a banana and make new banana plants. Instead, the banana plants are propagated and you get genetically identical clones that are making the bananas. Well, this was always the case, and the bananas that we had, there was a fungal disease. Okay, a fungal disease that was introduced to the bananas. Now, if everything is identically the same, everything is genetically the same, the resistance for that disease is the same. And in this case, the resistance for that disease was very, very low, which means that that fungal disease was able to very quickly wipe out that entire lineage of bananas, and they have since gone extinct. Okay, when we're talking about a changing environment, so, changing weather, new diseases, different sorts of, rain patterns, whatever it may be, temperatures, we're going to start to see individuals who rely on asexual reproduction either having low survival or having the ability to adapt very quickly to that change. So like bacteria, for instance, right? Bacteria can adapt very quickly to a change because they do reproduce through mitosis. And individuals who happen to have a mutation and a resistance to, say, an antibiotic will be the ones who then are making those genetic identical clones of themselves, okay? Meiosis, on the other hand, while it gives us unique genetic recombination and halves the number of chromosomes, resulting in haploid gametes, it's a relatively slower process, generally speaking. If I say a bacteria reproduces asexually and humans reproduce sexually, there's a very big difference in generational times, and we'll talk about that a little bit more in evolution. This does often result then in high survival in a changing environment because individuals are more likely to have variations that will allow them to adapt to that change. And so, again, all of this is really within whatever context you look at it, because if we look at, like I said, bacteria, it's not quite the same thing, where meiosis can be really beneficial. A lot of organisms do both. Actually, most organisms don't rely on meiosis as their only form of reproduction. Most plants and fungi and protists actually do mitosis and will make copies of themselves as much as possible. In fact, a lot of animals actually rely on that as well, which we'll talk about in our Diversity of Life unit 5. Okay, so keep that in mind. We're going to get into the nitty gritty of meiosis though. So we're going to talk about how we actually create these genetically unique haploid gametes. Okay, so when we talk about meiosis, we really look at it from the human perspective. If you were to take like a Bio 112 with me down the road or other biology classes in the future, you might find out that this is not the norm for meiosis. This is not the norm for sexual reproduction in most organisms. But for our purposes in Bio 110, we're going to talk about it in a way that we can easily grasp and understand, okay? We as humans are adults, and adults are typically diploid, and you're going to have male and females. In biology, males produce sperm and females produce eggs, okay? So if you are a biological male who is XY chromosomes, you will produce sperm, and if you are a biological female who is XX, you will produce an egg. Now, again, there's a lot of variation in here, and if you do have more questions about that, let me know. We can talk about it. I think I like to throw a discussion board or something out there related to this because there's a lot of individuals who don't fulfill that like biological requirement. So we're going to keep it at the surface level for Bio 110, okay? When we talk about an individual who is an adult, I am an adult female, okay? If I were to reproduce, I would produce an egg. My husband would produce a sperm. If that sperm and egg were to come together, we would have a zygote. The sperm fertilizes the egg, and that results in a zygote. A zygote is now a diploid cell because we took two haploid cells and put them together. That means that our child would be 50% my DNA, 50% my husband's DNA, okay? But what 50% is completely randomized by the process of shuffling chromosomes and crossing over, okay? And we'll talk about those a little bit more in a couple slides. What's really cool though is if you have siblings, okay, I have siblings. My brothers, there are some baby pictures that my mom didn't label of my brothers, and we don't really know who is who. There are some baby pictures and you go, I think that's Brandon. Maybe it's Kyle. I don't know. It's one of them, okay? And I look a lot like my brothers. We all look very similar to one another, especially when we were younger. You know, even though we only share 50% of our DNA. We share parents, okay? They're not my half siblings, they're my full siblings. But because fertilization produces new combinations, we actually end up with the most ridiculous number of chromosomally different zygotes possible, even assuming no crossing over, which is one of the ways that genetics are reshuffled in an individual before fertilization happens. So when I talk about how, you know, we are genetically unique, that's what I mean. That being said, some people say copy paste. My husband said copy paste to his son. They look, they act, they seem similar. I'm sure if we had had children, it would have been the same sort of thing. I never wanted kids. I have my stepson. I love it. I didn't want to go through this whole process here, but it is fascinating, right? And once that zygote is formed, then the cell goes into mitosis, and that mitosis will result through the entirety of the life, and then we will have some cells that are capable of being a sex cell later in life when we reach sexual maturity, okay? Now, I had mentioned about twins. Now, we are going to be really, really unique. If you are a fraternal twin, that means that you are the result of two different eggs being fertilized by two different sperm. You are no more similar to one another than any other set of siblings, okay? Because two eggs were released, both eggs were fertilized, and then those two embryos develop at the same time. So they're fraternal twins. They're not identical. They are fraternal. And they're normal siblings in that genetic combination there. But what about identical twins? Well, identical twins are the result of a single fertilization. So one egg and one sperm are going to fertilize and create one zygote. But early on in that mitosis, that division of the zygote into a blastocyst, into a bunch of cells, there can be a split where those cells don't really stick together. If those cells split and continue to grow, that's when you'll get identical twins, and that's why they share their genetic material. Identical twins are the result of a single fertilization event. Fraternal twins, two fertilization events, you're going to have a random combination there, which is why some fraternal twins can look incredibly different, or why you can have a boy and a girl, and you can have that male and female kind of chromosome mixing up. But identical twins, you'll have two males, two females, triplets. Okay, I have to just throw this out there because I think this is fun. If they're not identical triplets, generally you have a set of identicals and a fraternal. And so you had 2 fertilization events, and one of those fertilization events became identical twins and split during that early mitotic division. So if you've ever wondered about twins, that's what that's about. I just think it's fun. I had a class one semester where we were talking about this, and then another student will be like, well, I'm a twin, so what about this? And I'm a twin. I think 8 of them told me they were twins in that class, and I was convinced that they were pranking me. It was crazy, but now we understand why, okay? So let's talk about these chromosomes and how we can see crossing over and independent assortment occur. So when we talk about chromosomes, okay, remember a single chromosome is going to replicate itself, okay, and then it has those 2 identical sister chromatids that are held together by a centromere. But when we say that we as humans have 23 pairs of chromosomes, what we're saying is that we have homologous pairs. We have chromosomes who have similar size, shape, and centromere location, and as such contain the same genes for the same traits, even though the variety of those genes may be different, okay? So in this image, I have 1 chromosome that's green, 1 chromosome that's purple, and while those chromosomes replicate during the S phase of interphase, just like we would see in mitosis, we are going to see that these homologous chromosomes are going to find one another, and that's going to be a big part of meiosis that's different from mitosis. If sister chromatids were to exchange genetic information, nothing would be different. But having homologous chromosomes means that when we have this mixing up that occurs, this is why. You inherit 1 chromosome from mom and 1 chromosome from dad. So you inherit a female and a male copy of each homologous chromosome. And we know that they're homologous because if we look at something called a karyotype, which is a picture of someone's chromosomes, we can generally match them by shape, by length, and by centromere position. So chromosome pair 1, and if you're not colorblind, the staining, it's a little bit funky if you are colorblind, I apologize. But you can see based on the length and centromere position. The chromosomes are stained in such a way too that we can actually see them matching based on the colors. And so chromosome pair 15 has a little side that's green and then long arms that are pink. And then this particular individual happens to have 2 X chromosomes. Since this individual has 2 X chromosomes, we know that this individual is biologically female and would likely produce eggs. Okay, and here we have chromosomes pairs 1 through 22. Pairs 1 through 22 are considered autosomes, and pair 23 in humans is called the sex chromosome. Now again, other animals are going to have other variations on this. I think chickens are XO. There are some insects that are Z something. They have different chromosome names, but they're going to still have chromosomes that identify them as male or female. Okay, so now that we know some of the big vocab here, let's go through the basic steps of meiosis and kind of talk through what's happening here, and then we'll compare that to mitosis. So in meiosis 1 and 2, okay, we're going to see that so in meiosis, we have to talk about it as meiosis 1 and meiosis 2. In order to get 4 cells at the end, we have to actually see 2 divisions. The first division creates 2 cells, and then those 2 cells go through division a second time to generate 4 cells, okay? If you do not have the numbers, if you do not say meiosis 1, if you do not say meiosis 2, if you are saying prophase and you don't say prophase 1 or prophase 2, you're talking about mitosis, okay? Or you're talking about meiosis in general. So these numbers do matter because in meiosis 1 is where we see all of the unique events for meiosis as compared to mitosis. So in meiosis 1, our homologous pairs line up with one another. They find their match, okay? At that point, they are going to then separate from one another. And that is how we end up with 2 cells that are haploid. So our cell is diploid. In this case, we have 4 chromosomes. Those homologous pairs have found their match, right? Their similar length, shape, centromere position. And then they are pulled apart into separate poles. And so our new cells will have 1 copy of each chromosome, okay? That is what makes them haploid. After that, then meiosis 2 is very similar to mitosis in that meiosis 2 is going to go through divisions of the centromere and the sister chromatids, resulting in daughter nuclei, and we get 4 gametes at the end. Okay, so meiosis 1, everything unique happens. Meiosis 2, same steps as what we see in mitosis. And both actually have prophase, metaphase, anaphase, and telophase. We just need to consider that they're like 2 separate phases here. Okay, so let's talk about crossing over, and then we have a check your understanding. All right, crossing over is going to occur during meiosis 1, during prophase 1. And I had mentioned that sister chromatids, if they were to exchange segments of DNA, it wouldn't matter because they're identical to one another, right? The sister chromatids are copied, held by their centromere. They're identical to one another. Swapping any sort of information wouldn't do anything. However, when our homologous pairs find one another, okay, what they can do, they don't have to, and this can be variable, which can obviously then increase the randomness of genetic variation, is they can actually have their non-sister chromatids cross over. Where those non-sister chromatids touch is called a synapsis. Okay, that synapsis will actually result in an exchange of genetic information. So when my non-sister chromatids exchange genetic information, I actually now have chromosomes that have chromatids that are no longer identical. And we call this recombinant DNA. So this DNA has been recombined, right? The homologous pairs have exchanged information. So for instance, say that this first chromosome had a gene that coded for blue eye color, and this red chromosome had a gene that coded for brown eye color. Well, on my sister chromatids, both of those genes would be for blue eye color, and on the red, or this other chromosome, both of those would be for brown eye color. But now they're mixed up, and so I actually have one chromosome on the blue pair, on this first pair, that would carry the blue eye gene, but now the other one carries the brown eye gene. They're no longer identical to one another. Okay, they're recombined. This is recombinant DNA. And then the same thing on the next chromosome, which gives me 4 different chromosomes because the chromosomes are no longer identical on their sisters. But you can only see this exchange happening between the non-sister chromatids. Again, if I exchange sister chromatid sections, it wouldn't change anything. Okay, I strongly recommend like going through and drawing this out or getting some Play-Doh and like playing with this with Play-Doh. You should have pop beads in your lab kit. Maybe try and play around with those. But just so you know that this is really important because this process results in that recombined DNA that gives us 4 unique chromosomes. So if I were to put each one of these chromosomes into a cell, I now have 4 new haploid cells, and I wouldn't even have to do anything else, okay? Here is your first check your understanding. Do make sure that you can answer these and answer them fully, okay? Make sure that you can answer them fully, and then we're going to get into the nitty-gritty of those phases of meiosis. All right, so meiosis has the 2 divisions, right? Meiosis 1 and meiosis 2. Everything unique happens in meiosis 1. Meiosis 2 is most similar to mitosis, but you still need to include the 1 or 2. I don't care if they're Roman numerals or Arabic numerals. Doesn't matter to me, okay? However you want to write it. But if you talk about prophase, it's mitosis. If you talk about prophase 1, that's meiosis 1. If you talk about prophase 2, that's meiosis 2, okay? We still see PMAT because we're still going through that process of dividing the genetic material into new nuclei. But now we're trying to get to 4 haploid nuclei rather than just the 2 identical nuclei we see in mitosis. So, meiosis 1. Okay? In meiosis 1, in prophase 1, the nuclear envelope dissolves, which again we need to do if we want to have access to that genetic material. The nuclear envelope has to dissolve. But when the nuclear envelope dissolves, we don't see chromosomes just in there. We actually see these tetrads where these homologous pairs are linked together. They're either just hanging out next to each other, or we could see the non-sister chromatids crossing over at this point. So crossing over occurs at prophase 1 of meiosis 1. it makes sense, right? Because if I want to carry through that recombined DNA through the rest of the steps, I need that to happen early on so that it can actually follow through, okay? So prophase 1, crossing over occurs, homologous pairs line up together, they kind of find each other. We call this a tetrad because then there's 4 arms, so there's a tetrad, okay? And then once that's all good to go, we move into metaphase 1. In metaphase 1, okay, your tetrads are still together. Those homologous pairs are still hanging out with their buddy. Now, those homologous pairs are going to line up at the middle of the cell. However, what we need to keep in mind here is that the homologous pairs can line up however they want in the middle of the cell, so long as they're with their buddy. What does that mean? I think the easiest way to think about this is when I was a student, when I was in kindergarten, my teacher always had us line up in 2 lines, right? Boys on one side, girls on the other, and we had to line up alphabetically. I don't know. My kindergarten teacher was she was awesome, but she was also very controlling, okay? So we had to line up in that same way every single time. Well, that is not independent assortment. That is a dependent assortment, right? I am a girl. My last name was Wiggins. I was at the end of the line every single time. I don't think anybody was ever after me. And I lined up next to the boy whose last name was Zimmerman. That is just how it worked, because that was the way that the alphabetical order of those 2 lines worked. But if instead of having your students, your kindergartners line up in 2 lines in a certain way, you just told them to line up in 2 lines. Every day when you told them to line up in 2 lines, especially if they're kindergartners, they're going to get in whatever order they want to, so long as they're in 2 lines, who cares, right? That is what metaphase 1 is like, except for they have a buddy, so they have to stand next to their buddy. But their buddy can shuffle. You can be either on the left or the right line, or you can be anywhere along the line. It doesn't matter, but you're going to be in those 2 lines. That is independent assortment. So your homologous pairs are going to find their buddy, and they're going to line up in the middle, and then that middle could shift. It could be every other one is a different, chromosome, like from mom or dad, and it can be all mixed up, and that's okay, as long as they're with their buddy. Because at that point, we are going to separate those homologous pairs. Once we move into anaphase 1, your homologous pairs separate. And so instead of having this, 2 line of chromosomes in the middle, you are now going to have X's pulling to the opposite poles. And again, if we have our recombined DNA, that recombined DNA is now pulled to opposite poles. And we end up with a unique combination of those chromosomes on either end of the cell. Once those chromosomes are pulled to the opposite ends of the cell, the nuclear envelope may or may not reform, because we're going right into meiosis 2. And like we talked about with photosynthesis, if you made G3P and you need to use it right away in cellular respiration, you're not going to make glucose to then literally turn around and break glucose. You're not going to make a nuclei, like a nuclear envelope, to turn around and break a nuclear envelope, which is what the next step would be. So we might see it forming, we might not. It really depends on the cell and at what point during reproduction and this we see. Sperm, for instance, I'm pretty sure don't reform this, but eggs might, At this point, our cells will go through cytokinesis because our goal is 4 cells, This isn't one of those ones where we see multinucleated. We do not want multinucleated gametes. We are going to see cytokinesis occur here, either through the cleavage furrow in animal cells or cell plate in plant cells. And our nuclei are now haploid. So we started with 4 chromosomes in the first cell in the parent cell, and we ended up with 2 chromosomes in each of the new daughter cells. And so we actually do see that division of the chromosome number into half the number and recombined DNA. Everything that's unique about meiosis has happened in meiosis 1. At this point, prophase 2 is going to break down the nuclear envelope if it formed. Metaphase 2, we're going to see the sister chromatids attached to the centromere, aligning in the center. Anaphase 2, the sister chromatids will separate and move to opposite poles. And then telophase 2, we have 4 nuclei form around haploid cells that are haploid genetics that are unique. And our cells will separate through cytokinesis, resulting in 4 unique haploid daughter cells called gametes. everything that we see in mitosis is happening here, but we have 2 cells going through each step rather than just the one. You will be asked to look at cells, like these images that aren't labeled, and tell me if that cell is in mitosis or meiosis and support your reasoning. So do make sure that you can do that, because that is a common question on the exam. Okay? When we compare meiosis with mitosis, what we see is, again, meiosis, everything unique happens in meiosis 1, and then meiosis 2 is simply the division into the 4 cells, whereas mitosis, we are simply dividing into 2 identical cells to the parent cell. We still start with the same chromosomes, especially if we're talking about the same individual with the same genetics, and like my skin cells, remember, I want my DNA and my skin cells to be the same. But in my gametes, I would want genetic diversity. Genetic diversity can increase survival rates in populations. Okay? So those are the steps of meiosis. Make sure you can do them. I know I have a cool worksheet in Canvas that is something that you can just fill in from these images. I suggest doing it with color and doing it for mitosis and for meiosis using the same colors so that you can identify the similarities and differences there. Okay? When we look at mistakes then, what happens if non-disjunction is a mistake, but what happens if meiosis has a mistake? You could still see cells going through apoptosis as a result, because if the cells don't have the right things to cross those checkpoints during interphase, if there's damage to the DNA, if the cell has a problem, a lot of times those cells will still go through apoptosis. But one of the things that we see that's the most common with mistakes in meiosis is something called non-disjunction. Non-disjunction is a failure of the chromosomes to separate. Now, there are 2 times that this can occur because the chromosomes separate at 2 different points. They can separate during anaphase 1 of meiosis 1 or during anaphase 2 of meiosis 2. Now, looking at these 2 images, I want to give you a minute to think to yourself which one would result in more problematic zygotes. So which one, if the cells were fertilized, are going to have more zygotes with the wrong number of chromosomes? Okay, hopefully you thought about it for a second. And hopefully your answer was that if non-disjunction occurs during anaphase 1 of meiosis 1, we have a bit more of a problem. Because at the end of the day, our gametes, not a single one of our gametes will have the correct haploid number of chromosomes. Instead, 2 of our gametes would be 2n minus 1, meaning they'd be missing a chromosome, and 2 of our gametes would be 2n plus 1. If this gamete were to be fertilized, any of these gametes were to be fertilized, our individual zygote would be 2n plus 1 or 2n minus 1. And in most cases, that is not good, okay? If you have 3 copies of chromosome pair 21, for instance, you would find someone with Down syndrome. And that is likely to occur if we have these extra chromosomes. You don't really hear about people missing chromosomes because you do need all of your genetic material, and missing chromosomes can be way more of a problem. In non-disjunction during meiosis 2, we actually end up with 2 gametes that would be a normal haploid number. And we would get 2 gametes. One would be n plus 1 and one would be n minus 1. If either one of those were fertilized, we would see an abnormal chromosome number. But if either of the other 2 are fertilized, we probably wouldn't know that a non-disjunction event ever occurred. These are still failures to separate. They can result in apoptosis, but more often than not, if they do get fertilized, if there is a fertilization event of a cell that went through non-disjunction, we do see individuals having spontaneous abortion, also known as miscarriage. If you have an abnormal number of chromosomes, generally there's going to be some sort of problem with development of that zygote early on in its development. And some people end up not even knowing they were ever pregnant because of these events that occur, and it can occur so early, and it's just not conducive to life. If you are missing one of chromosome pair number 1, or if you have an extra of chromosome pair number 1, that is a failure to thrive. There's no way that that individual is going to develop fully and appropriately. In fact, there are only 3 copies of chromosomes in humans that you can have a full extra pair of. That would be chromosome number 13, 18 and 21, as well as your sex chromosomes. Sex chromosomes are different. We're going to talk about those, okay? So here's real quick. Check your understanding. We're going to talk more about some of those monosomies and trisomies, but make sure that you can understand these, okay? Make sure that you can go through this. And let's get into it. So, non-disjunction has occurred. What actually is happening here? Well, if we're talking about something like trisomy 21, where you end up finding an individual who has a third copy of chromosome number 21, it can be a complete copy or even a partial copy of chromosome 21 if that chromosome broke while it was pulling apart. You do see individuals that are going to have the characteristic symptoms of Down syndrome. Now, this is not always going to be a big problem. Some people have, it's a range, but we do tend to see individuals having more learning difficulties. Oftentimes they have some more issues with heart and gastrointestinal things, and it can cause some issues for them long term. But anyone who's met anyone with Down syndrome knows that they're the kindest, nicest, most wonderful people who just want to love and be loved. That's not to say that it can't result in a non-viable pregnancy, because there can be some very serious complications depending on which genetics you inherit on that chromosome. I also said that you could potentially have an extra copy of 13 or 18. Both of those are really, really problematic, and that child is going to have a lot of times needing a feeding tube, a ventilator, life support. It's still not really conducive to life at that point. What I want to point out from this karyotype is that this individual has sex chromosomes X and Y, which if we look, sex chromosomes are a little bit different than the rest of our autosomes, because while our autosomes have these good, clean, homologous pairs, the sex chromosomes do not. If you had two X's, those would be considered a homologous pair. But in this instance, the Y chromosome is so tiny compared to the X that it is not considered a homologous pair. And one of the things that I do want to point out now that we'll talk about a little bit more with sex-linked genetic disorders, the Y chromosome has 200 genes. The X chromosome has over 2,000 genes. You need at least one X chromosome for life to occur. If someone were to inherit a single Y chromosome, that is also one of those situations that will result in a spontaneous abortion, because an X chromosome is responsible for your inner ear, your jaw, your lungs, your heart, parts of those. You absolutely need that chromosome with the genes on it for that. You don't need two copies though. The second copy could be a Y, and that Y will just denote maleness. There are genes on there that are responsible for testosterone reception. Remember our hormones and whether or not you can receive a lot of testosterone or not. And that actually leads us to our last little kind of what's weird here, and this can be when we see some abnormal sex chromosome numbers. Now, this is more common, and these individuals are considered to be intersex. So an individual who has too few or too many X or Y chromosomes can have a relatively normal life. And when I was talking about the karyotype earlier, or the XX and XY, this is what I mean. We can have genes that are going to receive certain hormones differently than others. The Y chromosome generally denotes maleness. And so we can see sometimes individuals born who are XO, so they just have a single X, so this is a monosomy situation, and this is someone we would consider to have Turner syndrome. A lot of these individuals are sterile and won't find out necessarily, that they're sterile until later. Not all of them are. That's not always the case, okay? A lot of them do tend to have issues with inner ear, with heart and lung development. And if you're really curious about some of these abnormal sex chromosome differences, there is an episode of this podcast, Will Kill You, on Turner syndrome, which is fantastic because they discuss this with a lady who has Turner syndrome. She's fantastic. There is also a really good show on Netflix called You Can't Ask That. It's an Australian show, and they do have an Australian episode on intersex individuals, and it's absolutely fascinating because you can have individuals who are XO and consider to have Turner syndrome. You can have individuals who are XXY and have something called Klinefelter syndrome, and while they are then determined to be biologically male, that does not mean that they actually are able to receive testosterone, and so they may end up displaying as biologically female with their secondary sex characteristics. Some individuals with Klinefelter syndrome are not sterile, and in that episode on Netflix from You Can't Ask That, absolutely that is one of the things that they talk about. And there's a huge array of this. We're finding out with humans that our sex chromosomes are really like arranged all over the map. Part of that episode of This Podcast Will Kill You actually talks about the history of X and Y chromosomes and like where they came from and how we understood them. A lot of this sort of situation is something that is very personal, is very like individual to that person and isn't necessarily anyone's business. But you can have multiple copies. There have been individuals who are XXX. Do not Google that. I will never let this student forget this. I have an adult student in the past who I was teaching this, and he was like, well, I was really curious about trisomy X females. And so I went home and I Googled XXX females. And then I had some explaining to do to my wife. Sir, that is not my fault. I said trisomy X, not triple X, okay? There have been individuals who are XXYY or XXXY. Sex chromosomes often do, have various non-disjunction events that occur and also various genes that actually function or don't function on there. I have seen some interviews with people who are biologically, they thought they were male, they were raised male, they're intersex, they do a genetic test, it turns out that they were XX, but they were raised male. So we can see that there's a huge range here. If you're really curious about it, definitely check out some of those things that I had mentioned, okay? Make sure that you can check your understanding. Let me know if you have any questions. As always, make sure you can answer all of these. Any of these questions are fair game on exams. Thank you very much.